Okay, good morning. I'm discussing ion exchange chromatography. Ion exchange chromatography is a process by which a mixture of similar charged ions can be separated by using an ion exchange reagent which exchanges ions according to their relative affinities. It is a process that allows the separation of ions in polar molecules based on their charge, a form of liquid chromatography in which the stationary phase is an ion exchange reagent, which may be cation exchange reagent or anion exchange reagent. It can be used for almost any kind of charged molecules, including large proteins, small nucleotides, and amino acids. Also, we can define ion exchange chromatography is a process by which a mixture of similar charged ions can be separated by using an ion exchange reagent which exchanges ions according to their relative affinities. Principle of ion exchange chromatography. It is a chromatographic process that separates ions in polar molecules based on their affinity to their ion exchanger. The process of removing observed ions is known as elution. The solution employed for elution is called eluent, and the solution obtained from elution is called eluate. The liquid flowing into another, that is the liquid entering the ion exchange column, is called effluent, and liquid leaving the column conveniently is called effluent. Influent and effluent. The liquid which into the another and the liquid entering the ion exchange column is called influent, and liquid leaving the column conveniently is called effluent. See, a reversible exchange of ions between the ions present in solution and ion exchange resin, cation exchange chromatography. Positively charged proteins are reversibly altered to immobilize negatively charged base polymers. Anion exchange chromatography. Negatively charged proteins are reversibly altered to immobilize positively charged base polymers. Just opposite. See, positively charged proteins are reversibly adsorbed to immobilize negatively charged base polymers. And just opposite, anion exchange chromatography. Negatively charged proteins are reversibly adsorbed to immobilize positively charged base polymers. Cation exchange, anion exchange, also we can write in the diagram. Cation exchange reagents, cation exchangers. The cation exchangers have negatively charged with positively charged mobile counter ion available for exchange. Example, sulfonic acid, strong cation exchanger, carboxylic acid, weak cation exchanger, cation exchange resin. A typical example is displacement of mobile ions from DOX50 or sulfonated polystyrene cation by calcium ions. By passing a solution containing sodium ions, the calcium ions may be removed from the resin and the original sodium form of resin is generated. Figure sulfonated polystyrene cation exchange resin. Anion exchange resin. The anion exchangers have positively charged exchanger with negatively charged mobile counter ion available for exchange. Quaternary ammonium polystyrene, anion exchange resin. Anion exchangers are ionized in both hydroxide in salt form. Figure structure of anion exchange resin, quaternary ammonium polystyrene. Anion exchanger versus cation exchanger. They contain immobilized cationic groups that bind to the anions. Anion exchanger contain immobilized cationic groups that binds to the anions. The most common matrix with attached diethyl aminoethyl. Cation exchangers, they contain immobilized anionic groups that binds to the cations. Example of matrix with carboxymethyl groups. Application of ion exchange chromatography. Ion exchange chromatography is used for the suppression of rare earths, amino acids, carbohydrates, proteins, transition elements, complex ions, and even isotopes. Separation of lanthanides. Ion exchange method is most successful and rapid method for the separation of lanthanides. The techniques which is used for the separation of lanthanide ions with the help of cation exchangers is known as cation exchange chromatography. In this method, a solution of lanthanide ions is run down a column of synthetic ion exchange resin being sulfonic group. The lanthanide ion replaces the hydrogen ion in the sulfonic group. Thus, the lanthanide ions get fixed to the resin and hydride ions get washed down to the column. Then a buffered solution of citric acid, ammonium citrate at pH 5.5 is passed through the resin. The solution elutes the lanthanide plus the ions from the resin one by one. The lanthanide ions are complex as the citrate complex. Therefore, these smaller ions come out of the column first. One by one separation is possible because the ions move out of the column at different rates. The solution flowing out of the column is collected as different portions in different containers. Each container receives a solution portion which is reached a particular metal. Sorry. On adding the ammonium oxalate solution to each solution portion, the metal ion is precipitated as its oxalate ion, which on heating produces oxides. Removal of interfering radicals. Phosphate ions interferes in the stimulation of calcium and barium ions by oxidate method. Removal of ion is done by passing a solution of calcium and barium ions through a sulfonic cation exchanger. The calcium and magnesium ions held by the resin will be removed by using suitable eluent and estimated by usual methods. Softening of hard water, calcium and magnesium and other divalent ions are removed by passing hard water through cation exchanger charged with sodium. The calcium and magnesium ions are returned in the resin, whereas sodium ions pass into the solution. Demineralization of water. Water is the first pass through an acidic cation exchange where cations are exchanged by hydrogen ion. It is then passed through the basic anion exchangers to exchange chloride, sulfate, hydroxide, ions of the resin. Determination of anion. This technique is used for the separation and determination of anions like chloride, bromide, nitrate, sulfate, etc. Protein purification and exchange chromatography is commonly used to purify proteins from complex mixture by selecting the appropriate stationary phase and elution condition. Specific proteins can be separated and purified by high purity and yield. Pharmaceutical industry in drug development and exchange chromatography is employed to separate and purify active pharmaceutical ingredients, ensuring high quality and safety of medication. Environmental analysis. This technique plays a crucial role in water and soil analysis by separating and quantifying ions, heavy metals, and other pollutants, aiding in environmental monitoring and regulatory compliance. Nucleic acid separation and exchange chromatography is utilized for the separating DNA and RNA fragments based on their charge characteristics. This is essential in molecular biology research and genetic analysis. Biotechnology. Ion exchange chromatography is often a part of downstream processing in biotechnology, helping to isolate and purify biomolecules like enzymes, antibodies, and hormones. Okay, it's all about ion exchange chromatography. Thanks for watching and request you all like, comment, share, and subscribe. Thank you for patience. That's now bye bye. See you again.